Time moves through us like water through stone, carving channels we call wrinkles, wearing away the sharp edges of youth until something softer, more weathered emerges. We watch our hands change, notice new lines around our eyes, feel our bodies respond differently to the world around us. Yet beneath these visible transformations lies one of biology's most profound mysteries. Why do we age at all? And could we ever learn to slow this ancient dance with time? Hi, I'm Luna, and if you find comfort in quiet curiosity, gentle science, and stories that drift like starlight, you're in just the right place. So wrap yourself in something warm, settle in. And if you'd like to hear more sleepy wonderings, feel free to like and subscribe because tonight, we're exploring one of humanity's oldest questions, a question that takes us from the tiniest molecular machinery inside our cells to the vast evolutionary forces that have shaped every living thing on Earth. The poetry of cellular time. To understand aging, we must first journey inward, past skin and muscle, past the familiar architecture of organs and bones, down to the microscopic world where life unfolds in its most fundamental forms. Here, in the quiet chambers of our cells, time keeps a different kind of record. Picture, if you will, the interior of a single cell as a bustling city that never sleeps. Streets of protein filaments carry cargo from one district to another. Factories called ribosomes work around the clock, assembling the molecular tools that keep everything running. The mitochondria, those ancient bacterial partners that chose to live within us billions of years ago, hum with the constant work of energy production, their membranes flickering with the gentle lightning of biochemistry. But cities age, and so do cells. The streets develop potholes. The factories begin to make occasional errors. The power plants start to produce not just energy, but also toxic byproducts that accumulate like smog in the cellular atmosphere. This is aging at its most basic level, not a single catastrophic failure, but a gradual accumulation of tiny imperfections, like a symphony slowly falling out of tune. At the heart of every cell lies its most precious cargo, DNA. Imagine these genetic instructions as an ancient library, its volumes containing the complete blueprint for a human being. But libraries require maintenance, and librarians grow tired. Each time a cell divides, creating two cells from one, the entire library must be copied. And with each copying, small errors creep in. Most of these errors are caught and corrected by molecular proofreaders, enzyme systems so sophisticated they put human editors to shame. They patrol the newly copied DNA like careful scribes, checking for mistakes, fixing what can be fixed. But some errors slip through, accumulating over time like dust settling on unused books. These mutations don't usually cause immediate problems. Our genetic library has remarkable redundancy built in. But slowly, subtly, they begin to change how the cellular city functions. Then there are the telomeres, perhaps the most poetic of all cellular timekeeping mechanisms. Picture the protective plastic tips on shoelaces, and you'll have a sense of what telomeres do for chromosomes. These repetitive DNA sequences cap the ends of our chromosomes, protecting the vital genetic information within from the wear and tear of cellular division. But here's where the poetry deepens. With each cell division, telomeres grow shorter. They sacrifice themselves, piece by piece, to protect what matters most. It's as if each cell carries within it a slowly burning candle, and when the wax runs low, the cell can no longer safely divide. Some cells die. Others enter a state called senescence, a kind of cellular retirement where they stop dividing but continue to live, often sending out chemical signals that can affect their neighbors. Leonard Hayflick, the scientist who first discovered this cellular aging limit in the 1960s, was studying normal human cells in laboratory dishes when he noticed something curious. No matter how well he fed them, no matter how perfect their environment, the cells would divide only about 50 times before they stopped. This became known as the Hayflick limit, and it suggested that aging might be programmed into our very cells, written in the language of telomere length. But the story grows more complex when we consider the enzyme telomerase, discovered by Elizabeth Blackburn and her colleagues in the 1980s. This remarkable molecular machine can actually rebuild telomeres, essentially rewinding the cellular clock. Most of our cells have very little telomerase activity. It's turned down low, like a radio playing in another room. But some cells, stem cells, reproductive cells, and unfortunately cancer cells, keep their telomerase turned up high, maintaining their telomeres and achieving a kind of cellular immortality. This discovery opened up tantalizing possibilities. If aging is partly about telomere shortening, and telomerase can lengthen telomeres, could we theoretically extend human lifespan by boosting telomerase activity? The idea captures the imagination, but biology, as always, proves more nuanced than our hopes might suggest. Cancer cells, after all, are essentially cells that have learned to ignore the normal rules of aging and death. They divide endlessly, maintain their telomeres, resist the signals that tell normal cells when to stop. In some sense, they have achieved the cellular immortality we might desire, but at a terrible cost. They've lost their ability to cooperate with the larger organism, becoming selfish actors in what should be a symphony of cellular collaboration. This paradox hints at something profound about aging. It may not be simply a failure of biological engineering, but rather an inevitable consequence of multicellular life itself. Individual cells that never age might threaten the integrity of the larger organism. Perhaps aging is, in part, evolution's way of ensuring that cellular citizens remain good neighbors, willing to step aside when their time is done. As we drift deeper into our exploration of aging, we encounter one of the most counterintuitive ideas in biology: that aging might not be a design flaw, but rather an evolved strategy. This seems almost absurd at first glance. Why would natural selection, which favors traits that help organisms survive and reproduce, allow, or even promote aging and death? The answer lies in understanding evolution not as a force that wants us to live forever, but as a process that shapes organisms to be successful at reproduction. Once an individual has passed its prime reproductive years, evolution's spotlight dims. The genes that affect how we age after reproduction matters less to evolutionary success than the genes that help us reach reproductive maturity and successfully raise offspring. This leads us to what evolutionary biologists call the mutation accumulation theory of aging. Imagine that some genetic mutations have different effects at different life stages. Perhaps they boost early vigor and reproductive success, but cause problems later in life. Natural selection would favor such mutations because their early benefits outweigh their late costs. Over many generations, these late-acting deleterious mutations would accumulate in the genome, creating a genetic burden that manifests as aging. But there's an even more elegant evolutionary theory, proposed by George Williams in the 1950s and refined by many others since. Called antagonistic pleiotropy, this idea suggests that some genes might have multiple effects, beneficial when we're young, harmful when we're old. The classic example is testosterone in males. This hormone promotes muscle development, competitive behavior, and sexual attractiveness, all useful for reproductive success. But testosterone may also contribute to prostate problems and cardiovascular disease later in life. Evolution keeps such genes because their early benefits outweigh their late costs. These evolutionary perspectives help explain one of the most striking patterns in aging: the incredible diversity of lifespans across different species. Why do some bristlecone pine trees live for thousands of years, while mayflies die within hours of reaching adulthood? Why do some whales live for centuries, while mice rarely see their second birthday? The answers often relate to ecological pressures and reproductive strategies. Species that face high predation pressure or live in dangerous environments may evolve to reproduce early and age quickly. There's no evolutionary payoff to investing in longevity if you're likely to be eaten before old age becomes relevant. Conversely, species in stable environments with low mortality pressure may evolve longer lifespans, especially if extended parental care or accumulated knowledge provides reproductive advantages. Consider the differences between mice and bats. Both are small mammals of similar size, but mice typically live 2 to 3 years, while some bat species can live 30 years or more. The difference lies partly in flight. Flying allows bats to escape predators that would doom earthbound mice, reducing external mortality pressure and making longevity evolutionarily advantageous. Bats have evolved more robust DNA repair systems, better antioxidant defenses, and other longevity-promoting mechanisms. Then there are the creatures that seem to have cracked aging's code entirely. The immortal jellyfish, Turritopsis doania, can reverse its aging process, transforming from adult back to juvenile form when faced with stress or injury. Hydras, simple freshwater animals, show no signs of aging and may be biologically immortal under ideal conditions. Some clonal organisms, like certain plants and fungi, can theoretically live forever by continuously producing new growth while shedding old parts. But perhaps the most fascinating examples come from the animal kingdom's longest-lived species. The Greenland shark, swimming slowly through Arctic waters, can live for over 400 years. Some specimens alive today may have been born before the American Revolution. These sharks grow incredibly slowly and reproduce late in life, a life strategy that would doom most species, but works in the stable, cold environment of Arctic seas. Even more remarkable are the tortoises of the Galapagos and Aldabra, some of which have lived for over 200 years. These creatures seem to age so slowly that century-old individuals can still reproduce successfully. Their cells show remarkable resistance to cancer and extraordinary DNA repair capabilities. They're living laboratories for understanding how evolution can craft longevity when environmental conditions favor it. These examples illustrate an important point. Aging is not inevitable from a biological standpoint. Life has found ways to postpone, halt, or even reverse aging processes. The fact that humans age and die is not because biology cannot create immortal organisms, but because our particular evolutionary history has shaped us for a life strategy that balances growth, reproduction, and longevity in ways that were adaptive for our ancestors. This evolutionary perspective also helps us understand why aging affects different parts of our bodies differently. Our hearts, brains, and other vital organs that we cannot replace must be built to last a lifetime. But our skin, constantly renewed by stem cells, can repair itself remarkably well throughout life. Our immune systems, so crucial for surviving to reproductive age, are maintained quite well until later in life, when evolutionary pressure for their maintenance diminishes. Understanding the evolutionary basis of aging doesn't just satisfy scientific curiosity. It provides crucial insights for those seeking to extend healthy human lifespan. If aging is partly the result of evolutionary trade-offs, then understanding these trade-offs might help us find ways to tip the balance toward longevity without sacrificing the benefits that come with our current life strategy. As we continue our gentle exploration through the landscape of aging, we discover that time wears many different faces as it moves through our bodies. Aging is not a single process, but rather a collection of interconnected changes, each following its own timeline, each contributing to the larger symphony of temporal transformation that we experience as growing older. In the quiet laboratories of cellular biology, researchers have identified what they call the hallmarks of aging, a set of fundamental processes that seem to drive the aging phenomenon across many different species. These hallmarks read like a catalog of time's various tools: genomic instability, telomere attrition, epigenetic alterations, loss of protestases, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. Each of these represents a different way that time leaves its mark. Consider protestases, the cell's ability to maintain proper protein function. Proteins are the workhorses of cellular life, but they're also delicate structures that can be damaged by heat, chemicals, or simple wear and tear. Young cells are remarkably good at monitoring their proteins, fixing minor damage, and recycling proteins that can no longer be repaired. But as cells age, this quality control system becomes less efficient. Imagine a master craftsperson's workshop, where every tool is carefully maintained and properly stored. Over time, the craftsperson grows less attentive. Tools accumulate minor damage. Some are put away incorrectly. Occasionally, damaged tools are used without being repaired, creating flawed work that requires more tools to fix. Eventually, the workshop fills with partially broken tools, and the quality of work suffers. This is what happens with protestases during aging. The cellular workshop gradually becomes less organized, less efficient, less capable of producing the molecular tools that keep life running smoothly. Mitochondrial dysfunction offers another window into aging's mechanisms. These ancient bacterial partners that provide energy to our cells begin to show their age in various ways. Their DNA, separate from the DNA in our cell nuclei, accumulates mutations at a relatively rapid rate. Their membranes become less efficient at producing energy. Most tellingly, they begin to produce more reactive oxygen species, molecular fragments, that can damage nearby structures. It's tempting to think of these reactive oxygen species as cellular pollution, and indeed, they can cause problems. But the story is more nuanced than simple toxicity. These molecules also serve as signals, communicating between different parts of the cell and even between different cells. As mitochondria age and produce more of these signals, they may be sending increasingly urgent messages about cellular stress, triggering responses that can be both protective and damaging. The immune system ages in particularly complex ways, a process scientists call immunosenescence. Our immune cells, tasked with distinguishing friend from foe and remembering past encounters with pathogens, gradually lose their precision. Some immune responses become overactive, creating chronic inflammation that can damage healthy tissues. Other responses become underactive, leaving us more vulnerable to infections and cancers. This immune aging helps explain why older adults often experience what researchers call inflammaging, a state of chronic, low-level inflammation that contributes to many age-related diseases. It's as if the immune system's thermostat becomes miscalibrated, running too hot in some situations and too cool in others, never quite finding the balanced response that characterizes healthy young immunity. Perhaps most poetically, our stem cells, those cellular fountains of youth that can transform into many different cell types, begin to show their age as well. Throughout life, stem cells serve as a repair service for our tissues, producing new cells to replace those lost to injury or normal turnover. But stem cells themselves age, becoming less active, less versatile, and sometimes less discerning about when and how to divide. Picture a master gardener who has tended the same garden for decades. In youth, the gardener plants with enthusiasm, prunes with precision, and responds quickly to the garden's changing needs. Over time, the gardener's energy wanes, responses become slower, and sometimes poor decisions are made about what to plant where. The garden doesn't collapse, but it gradually becomes less vibrant, less resilient, less able to bounce back from seasonal challenges. Epigenetic aging represents one of the most fascinating recent discoveries in aging research. Our DNA sequence remains largely the same throughout life, but the pattern of which genes are turned on and off changes dramatically. These epigenetic modifications are like bookmarks in the genetic instruction manual, marking which sections should be read actively and which should be ignored. As we age, these epigenetic bookmarks shift. Some genes that should be active become silenced. Others that should remain quiet begin to express themselves inappropriately. The result is a gradual drift away from the epigenetic patterns that characterize youthful cellular function. Some researchers have developed epigenetic clocks that can predict biological age based on these methylation patterns, sometimes revealing that some individuals are aging faster or slower than their chronological age would suggest. But aging doesn't affect all parts of our bodies equally or simultaneously. Our skin, exposed to environmental stresses and constantly renewing itself, may show signs of aging decades before our internal organs experience significant decline. Our cardiovascular system, working tirelessly to pump blood throughout our bodies, may maintain function remarkably well into advanced age, while our joints, bearing the mechanical stress of a lifetime of movement, may begin to show wear much earlier. This differential aging helps explain why some 80-year-olds can run marathons, while some 40-year-olds struggle with joint pain. It's not just genetics or lifestyle, though both matter enormously. It's also the reality that aging is not a uniform process, but rather a collection of many processes, each affecting different systems at different rates. The brain ages in particularly complex ways, with some functions declining while others remain stable or even improve. Processing speed tends to slow with age, and working memory, our ability to hold information in mind temporarily, often becomes less efficient. But crystallized intelligence, our store of accumulated knowledge and skills, can continue to grow throughout life. The aging brain may become slower at learning new tasks, but often becomes better at integrating complex information and seeing patterns that escape younger minds. Sleep, too, changes with age in ways that can profoundly affect how we experience the passage of time. The deep, restorative sleep of youth gradually becomes lighter and more fragmented. We may find ourselves waking more frequently during the night, spending less time in the deepest stages of sleep, where memory consolidation and physical restoration occur most effectively. These sleep changes aren't just inconveniences. They may contribute to aging itself. During deep sleep, our brains activate a kind of cleaning system that flushes out cellular waste products, including some of the protein aggregates associated with neurodegenerative diseases. When sleep becomes less efficient with age, this cleaning process may become less effective, potentially accelerating some aspects of brain aging. As we venture deeper into the microscopic realm where aging unfolds, we encounter some of the most remarkable discoveries in modern biology. Here, in the space between molecules, we find the actual machinery that governs how time affects living things. These discoveries have transformed our understanding of aging from a vague sense of inevitable decline into a precise catalog of molecular processes that can be studied, measured, and potentially modified. One of the most significant breakthroughs came with the discovery of sirtuins, a family of enzymes that seem to function as longevity regulators within our cells. Named after the silent information regulator 2 gene first discovered in yeast, sirtuins have been found in virtually all living things, from bacteria to humans. These enzymes perform a variety of functions, but they're particularly good at sensing cellular stress and responding by promoting survival mechanisms. When cells face challenges, limited nutrients, DNA damage, oxidative stress sirtuins spring into action. They modify other proteins, changing their function in ways that help cells survive difficult conditions. In laboratory studies, activating sirtuins can extend the lifespan of yeast, worms, flies, and sometimes mice. This has led researchers to wonder whether sirtuins might be molecular guardians of longevity, evolved mechanisms that help organisms survive tough times, and, as a side effect, slow the aging process. The connection between sirtuins and longevity became particularly intriguing with the discovery that these enzymes can be activated by certain compounds. Resveratrol, found in red wine and the skin of grapes, was one of the first natural compounds shown to activate sirtuins. This discovery sparked tremendous interest. Could it be that the supposed health benefits of moderate red wine consumption, observed in some populations, might be due to sirtuin activation? The story became more complex as researchers dug deeper. While resveratrol does activate sirtuins in laboratory conditions, it's not clear whether drinking red wine provides enough resveratrol to have meaningful effects. And sirtuins themselves turn out to be responsive to many different signals, not just resveratrol. They're activated by fasting, by exercise, by various forms of mild stress that seem to trigger beneficial adaptive responses. This led to one of the most counterintuitive discoveries in aging research: the hormesis effect. The word comes from the Greek term for rapid motion or eagerness, and it describes the phenomenon where mild stress can actually promote health and longevity. It's as if our cellular machinery is designed to be strengthened by challenges, growing more resilient when faced with manageable difficulties. Exercise provides a perfect example of hormesis. When we work out, we create controlled damage to our muscles, stress our cardiovascular system, and challenge our cellular energy production. In response, our bodies don't just repair this damage, they overcompensate, building stronger muscles, more efficient hearts, and more robust cellular machinery. The stress of exercise triggers adaptations that make us more resilient to future challenges. Similarly, intermittent fasting periods of voluntary food restriction appears to activate many of the same longevity pathways that respond to natural food scarcity. When nutrients are limited, cells shift from growth mode to maintenance mode, activating repair systems, recycling damaged components, and generally becoming more efficient. Some researchers hypothesize that the constant abundance of food in modern life may actually accelerate aging by keeping cells in continuous growth mode, never triggering the maintenance and repair systems that evolved to help organisms survive lean times. The molecular pathways that govern these responses are incredibly complex and interconnected. The mTOR pathway, for instance, serves as a kind of cellular growth sensor. When nutrients are abundant, mTOR promotes cell growth, protein synthesis, and division. When nutrients are scarce, mTOR activity decreases, triggering autophagy, a cellular recycling process where cells break down and recycle their own components. Autophagy, whose discovery earned Japanese scientist Yoshinori Ohsumi a Nobel Prize, has emerged as one of the most important mechanisms for cellular maintenance and longevity. The word means self-eating, which sounds alarming, but describes a carefully controlled process. When cells are stressed or starved, they begin to digest their own damaged or unnecessary components, recycling the raw materials to build new, healthy cellular machinery. This process appears to decline with age, leading to an accumulation of cellular debris that can interfere with normal function. But autophagy can be stimulated by various interventions: fasting, exercise, certain medications, and even some natural compounds. This has led researchers to wonder whether maintaining robust autophagy throughout life might be one key to healthy aging. Another crucial player in the molecular machinery of aging is the insulin and insulin-like growth factor signaling pathway. This system evolved to help organisms respond to nutrient availability, promoting growth when food is abundant and conservation when it's scarce. But in the modern world, where food is constantly available and often high in refined carbohydrates that spike blood sugar, this pathway may be chronically overactivated. Studies in laboratory animals consistently show that reducing insulin and insulin-like growth factor signaling can extend lifespan. Mice with genetic mutations that reduce these signals often live 20–40% longer than normal mice. The mechanisms seem to involve improved cellular repair, enhanced stress resistance, and reduced inflammation. This connection between nutrient signaling and aging has profound implications for human health. It suggests that our dietary choices don't just affect our immediate well-being, but may also influence the rate at which we age. Diets that promote stable blood sugar levels, avoid chronic overnutrition, and occasionally trigger mild cellular stress through fasting periods might literally slow the biological clock. The discovery of cellular senescence has added another layer to our understanding of aging's molecular machinery. Senescent cells are cells that have stopped dividing, but haven't died. They accumulate with age, sitting in our tissues like broken-down cars on the cellular highway. But these cells aren't just inactive. They actively secrete inflammatory compounds that can damage nearby healthy cells and promote various age-related diseases. This discovery led to the development of a new class of potential anti-aging drugs called senolytics, compounds that can selectively kill senescent cells while leaving healthy cells alone. In mouse studies, removing senescent cells can extend healthy lifespan and reduce age-related diseases. Human trials of senolytic drugs are now underway, representing one of the most direct attempts to intervene in the aging process. The field of aging research has also been revolutionized by advances in understanding DNA repair mechanisms. Our cells possess sophisticated systems for detecting and correcting DNA damage, but these systems become less efficient with age. Some individuals are born with genetic variations that affect DNA repair capability, and these differences can have dramatic effects on aging and disease risk. People with defective DNA repair systems often experience accelerated aging syndromes, developing age-related diseases decades earlier than normal. Conversely, individuals with particularly robust DNA repair systems may age more slowly and live longer. This has led researchers to investigate ways to boost DNA repair capacity as a potential anti-aging intervention. All of these molecular discoveries paint a picture of aging as an incredibly complex but potentially modifiable process. Rather than being an inevitable result of wear and tear, aging appears to be governed by evolved regulatory systems that respond to environmental cues. These systems can be influenced by lifestyle choices, medications, and potentially future therapies designed specifically to slow aging. As our understanding of aging's molecular machinery has deepened, so too has our ability to imagine and attempt interventions that might slow, halt, or even reverse the aging process. We stand at a remarkable moment in human history, where the ancient dream of extending healthy lifespan is beginning to seem not just possible, but scientifically tractable. The first and perhaps most accessible approach to influencing aging lies in the realm of lifestyle interventions. The growing body of research on hormesis, the beneficial effects of mild stress, has validated many traditional approaches to healthy living while revealing the biological mechanisms that make them effective. Exercise, long known to promote health and longevity, is now understood to work partly through its ability to activate cellular stress response pathways. When we exercise, we create temporary metabolic stress that triggers adaptations throughout the body. Muscles don't just grow stronger, they also become more efficient at producing energy, better at clearing cellular waste, and more resistant to future stress. The cardiovascular system adapts by improving blood flow, reducing inflammation, and enhancing the body's ability to deliver oxygen to tissues. But perhaps most remarkably, exercise affects aging at the cellular level. Physical activity can increase telomerase activity, potentially slowing the cellular aging clock. It promotes the growth of new mitochondria, those cellular power plants that tend to become less efficient with age. Exercise also stimulates autophagy, the cellular recycling process that clears away damaged components and keeps cells functioning optimally. Different types of exercise may have different anti-aging benefits. Aerobic exercise excels at promoting cardiovascular health and mitochondrial function. Resistance training helps maintain muscle mass and bone density, both of which typically decline with age. High-intensity interval training may be particularly effective at triggering beneficial stress responses. Even gentle forms of exercise like walking, swimming, or yoga can provide significant anti-aging benefits, especially for individuals who have been sedentary. Dietary interventions represent another promising avenue for influencing aging. The discovery that caloric restriction can extend lifespan in many species has led to intense interest in understanding how food intake affects aging. While severe caloric restriction isn't practical or desirable for most people, milder dietary interventions may capture many of the same benefits. Intermittent fasting periods of voluntary food restriction appears to activate many of the same longevity pathways that respond to caloric restriction. During fasting periods, cells shift their focus from growth to maintenance, activating repair systems and clearing away damaged components. Some studies suggest that even short periods of fasting, practiced regularly, can have meaningful effects on markers of aging and age-related disease risk. The timing of eating may matter as much as the amount. Our cells operate on circadian rhythms, internal clocks that coordinate cellular activities with the day-night cycle. Eating late at night or at irregular times can disrupt these rhythms, potentially accelerating aging. Conversely, eating in sync with natural circadian rhythms may promote healthy aging. The composition of our diet also influences aging processes. Foods that promote stable blood sugar levels, provide abundant antioxidants, and supply essential nutrients for cellular repair systems may slow aging. The Mediterranean diet, rich in fruits, vegetables, whole grains, fish, and olive oil, has been associated with longer lifespans and reduced age-related disease risk. Some researchers hypothesize that this diet's benefits come from its ability to reduce inflammation, support cellular repair systems, and provide compounds that activate longevity pathways. Sleep represents another crucial factor in healthy aging, and one that's often overlooked in discussions of longevity interventions. During deep sleep, our bodies activate repair and maintenance systems that are less active during waking hours. The brain's glymphatic system, which clears waste products, including proteins associated with neurodegenerative diseases, is most active during sleep. Growth hormone, crucial for tissue repair and maintenance, is released primarily during deep sleep stages. As we age, sleep tends to become lighter and more fragmented, potentially reducing the effectiveness of these restorative processes. But sleep quality can often be improved through what's called sleep hygiene practices, like maintaining consistent bedtimes, creating a dark and cool sleeping environment, and avoiding stimulating activities before bed. Moving beyond lifestyle interventions, researchers are developing pharmaceutical approaches to target aging directly. Some of these build on natural compounds that appear to have anti-aging properties. Metformin, a diabetes medication that improves cellular response to insulin, has shown promise for extending healthy lifespan in laboratory animals and may have similar effects in humans. Large-scale clinical trials are underway to test whether metformin can slow aging in healthy individuals. Rapamycin, an immunosuppressive drug originally developed to prevent organ transplant rejection, inhibits the mTOR pathway that promotes cellular growth. In laboratory studies, rapamycin can extend lifespan in mice, even when treatment begins late in life. However, its immunosuppressive effects make it unsuitable for use as an anti-aging drug in healthy individuals, leading researchers to search for compounds that can inhibit mTOR more selectively. NAD+ boosters represent another promising class of potential anti-aging compounds. NAD+ is a molecule crucial for cellular energy production and DNA repair, and its levels decline with age. Supplementing with NAD+ precursors like nicotinamide riboside or nicotinamide mononucleotide may help restore youthful cellular function. While research in humans is still limited, studies in mice suggest these compounds can improve various markers of aging. The field of senolytics drugs that selectively eliminate senescent cells has moved from laboratory studies to human clinical trials with remarkable speed. Several compounds have shown the ability to reduce senescent cell burden in mice, leading to improvements in age-related diseases and extension of healthy lifespan. Early human trials suggest these drugs may be safe and effective, though more research is needed to fully understand their potential. Perhaps most ambitiously, some researchers are exploring whether aging itself could be treated as a disease. This represents a fundamental shift in how we think about aging, not as an inevitable part of life, but as a biological process that could be modified or even reversed. This perspective has led to investigations of more radical interventions, including genetic therapies to enhance DNA repair, stem cell treatments to restore tissue regeneration, and even attempts to reprogram aged cells back to more youthful states. The discovery that certain genetic factors can dramatically affect aging has opened the door to potential genetic interventions. Some individuals carry genetic variants associated with exceptional longevity. They may have more efficient DNA repair systems, better resistance to inflammation, or enhanced cellular stress responses. Understanding these genetic advantages could lead to therapies that provide similar benefits to those who weren't born with longevity genes. Stem cell therapies represent another frontier in anti-aging research, as our own stem cells become less active with age. Replacing them with younger, more vigorous stem cells might restore the body's regenerative capacity. Some experimental treatments involve harvesting stem cells from young donors or reprogramming adult cells back to a stem cell-like state. Even more speculative are attempts to reprogram aged cells directly, essentially resetting their biological age without changing their specialized function. This approach, inspired by the discovery that adult cells can be reprogrammed to become embryonic-like stem cells, has shown promise in laboratory studies. Researchers have managed to reverse some aspects of aging in mice by briefly activating reprogramming factors. Though much more work is needed to understand how this might be applied safely in humans. But with all these promising developments come significant challenges and ethical considerations. Many potential anti-aging interventions are still experimental, with unknown long-term effects. The complexity of aging means that interventions targeting one aspect of the process might have unintended consequences elsewhere. And there's the fundamental question of whether significantly extending human lifespan would be beneficial for individuals and society. As we consider these possibilities, we must also grapple with questions of equity and access. If effective anti-aging treatments are developed, will they be available to everyone? Or will they create new forms of inequality between those who can afford to slow aging and those who cannot? How would dramatically extended lifespans affect retirement, career progression, family structures, and resource allocation? These are not just scientific questions, but profound philosophical and social ones that will require careful consideration as anti-aging research progresses. The promise of healthier, longer lives is tantalizing, but realizing that promise responsibly will require wisdom to match our growing scientific capabilities. The philosophical dimensions of mortality as we near the end of our journey through the science of aging. We find ourselves confronting questions that extend far beyond biology and medicine. The possibility of significantly extending human lifespan, or perhaps even achieving biological immortality, forces us to examine fundamental assumptions about what it means to be human, what constitutes a life well lived, and how we relate to the passage of time itself. Throughout human history, mortality has been one of our defining characteristics. The knowledge that our time is limited has shaped our cultures, religions, philosophies, and individual life choices in profound ways. Art gains urgency from the artist's mortality. Love deepens when we know it cannot last forever. Achievements feel meaningful partly because they represent something accomplished within the constraints of a finite lifespan. If we could slow aging dramatically or halt it entirely, how would this change the human experience? Would extended lifespans be universally beneficial? Or might they come with unexpected costs to individual well-being and social functioning? Some philosophers and ethicists argue that mortality gives life meaning. The knowledge that we have limited time creates urgency and focus, pushing us to make choices about what matters most. If we had centuries or millennia ahead of us, would we become paralyzed by infinite possibilities? Would we lose the motivation to grow, change, and accomplish goals? Or would we simply find new ways to structure meaning and purpose across much longer timescales? There's also the question of whether extended lifespans would enhance or diminish human flourishing. On one hand, more time could mean more opportunities for learning, creating, experiencing, and building relationships. Imagine the expertise someone could develop over several centuries, the wisdom they could accumulate, the contributions they could make to human knowledge and culture. But extended lifespans might also bring challenges we haven't fully considered. Would relationships that currently last, till death do us part, remain sustainable over centuries? How would family structures evolve if multiple generations remained alive and active simultaneously? What would it mean for personal growth and renewal if the physical markers of life stages, childhood, adolescence, middle age, elderhood, were stretched across much longer periods. The social implications are equally complex. Many of our institutions and social structures are built around current human lifespans. Education typically occurs in youth, career building in early adulthood, peak productivity in middle age, and retirement in later years. If people remained physically and mentally capable for centuries, these life stage patterns would need complete reimagining. Economic systems, too, would face unprecedented challenges. Retirement savings, social security systems, and pension plans all assume people will work for several decades and then require support for a relatively brief retirement period. If retirement lasted for centuries, or if people needed to reinvent their careers multiple times across vastly extended lifespans, these systems would require fundamental restructuring. There are also questions about social mobility and innovation. In current society, new generations bring fresh perspectives, challenging established ideas and driving social progress. If lifespans extended dramatically, would established power structures become even more entrenched? Would the natural turnover that allows societies to evolve and adapt slow to a crawl? Yet perhaps these concerns reflect limitations in our current imagination rather than inherent problems with longevity. Humans have proven remarkably adaptable throughout history, creating new social arrangements as circumstances change. The transition from hunter-gatherer societies to agricultural civilizations, from rural to urban living, from pre-industrial to digital economies, all required fundamental shifts in how humans organize their lives and societies. Extended lifespans might catalyze similar innovations. People might develop new models for long-term relationships, perhaps involving formal renewals or transitions at regular intervals. Career patterns might become more cyclical, with individuals moving between periods of intense focus on particular fields and times of rest, exploration, or complete career changes. Educational systems might evolve to support lifelong learning in ways that keep centuries-old minds flexible and curious. There's also the question of whether extended lifespans would necessarily mean extended youth, or simply longer periods of aging. The goal of longevity research isn't just to add years to life, but to add healthy, vigorous years. If successful, anti-aging interventions would extend what researchers call health span, the period of life spent in good health, rather than simply prolonging frailty and decline. This distinction matters enormously for both individual well-being and social implications. A world where people remained physically and mentally capable for centuries would face very different challenges than one where people simply lived longer in states of increasing debility. The first scenario opens possibilities for extended creativity, productivity, and contribution to society. The second would create unsustainable burdens on healthcare systems and caregivers. From an environmental perspective, dramatically extended lifespans would also pose challenges. Earth's resources and ecosystems are already strained by current human population levels and consumption patterns. If people lived much longer while continuing to have children at current rates, population growth could accelerate dramatically. This might require new approaches to reproduction, resource use, or even space colonization to be sustainable. But longevity research might also contribute to environmental solutions. People who expect to live for centuries might be more motivated to address long-term environmental challenges, since they would personally experience the consequences of current environmental decisions. The long-term thinking that comes with personal longevity could drive more sustainable practices and technologies. The question of access and equality looms large in discussions of life extension. If effective anti-aging treatments are developed, Will they be available to everyone, or will they exacerbate existing inequalities? The prospect of a world divided between those who can afford to halt aging and those who cannot raises profound ethical concerns. Such disparities could create new forms of discrimination and social stratification. Imagine job markets where some applicants have centuries of experience, while others are limited by normal human lifespans. Consider the political implications of voting populations, where some citizens have lived through centuries of history, while others have only decades of experience. These concerns suggest that if life extension technologies are developed, ensuring equitable access will be crucial for maintaining social stability and justice. This might require treating anti-aging interventions as public goods, similar to how many societies approach basic health care or education. Religious and spiritual perspectives on aging and mortality add another layer of complexity to these considerations. Many religious traditions view death as a natural transition, part of a larger spiritual journey that extends beyond physical existence. Some see suffering and aging as opportunities for spiritual growth, ways of developing compassion, wisdom, and detachment from material concerns. How would dramatically extended lifespans affect these spiritual frameworks? Would they be seen as interfering with natural or divine plans, or as fulfilling human potential and divine gifts? Different religious traditions would likely respond in different ways, potentially creating new sources of cultural and spiritual conflict or evolution. From a purely secular perspective, there are questions about personal identity and continuity over vastly extended lifespans. What makes you you, your memories, your personality, your physical body, your relationships? Over the course of several centuries, all of these elements would change dramatically. Would a 500-year-old version of yourself still be meaningfully the same person as your current self? This touches on deep philosophical questions about the nature of personal identity and consciousness. Some philosophers argue that we're already very different people than we were decades ago, that personal identity is more fluid and constructed than we typically assume. From this perspective, extended lifespans would simply make more explicit the ongoing process of personal change and renewal that characterizes all human lives. Others worry that such extended change might threaten psychological continuity and well-being. There might be optimal lengths for human lives, long enough for meaningful accomplishment and relationship building, but not so long that personal identity becomes diffuse or life loses its sense of progression and purpose. These philosophical considerations don't have clear answers, and different individuals and cultures will likely reach different conclusions about the desirability of extended lifespans. Some people might choose life extension if it becomes available, while others might prefer natural lifespans for personal, philosophical, or spiritual reasons. What seems clear is that the development of effective anti-aging technologies would represent one of the most significant transitions in human history, comparable to the development of agriculture, written language, or industrialization. Like those earlier transitions, it would likely bring both tremendous opportunities and significant challenges, requiring careful thought and planning to navigate successfully. As we drift toward the end of our exploration through the vast landscape of aging and time, let us return to where we began, with the simple recognition that we are all travelers on a journey through time, our bodies and minds changing with each passing moment, like rivers slowly carving new channels through ancient stone. The science we've explored tonight reveals aging not as a simple march toward decline, but as an intricate dance between preservation and change, between the forces that maintain life and those that eventually transform it. At the cellular level, We've seen molecular machinery working tirelessly to repair damage, maintain order, and respond to challenges. We've witnessed the elegant mechanisms that evolved over billions of years to help life persist through countless generations, while also allowing for the change and adaptation that evolution requires. The telomeres shortening with each cell division, like candles slowly burning down. The mitochondria producing energy while generating the very reactive molecules that may contribute to aging. The sirtuins responding to cellular stress by activating ancient survival pathways. The complex interplay of genes, proteins, and environmental factors that determines how quickly or slowly we age. All of these represent life's ongoing negotiation with time, sophisticated biological solutions to the fundamental challenge of maintaining complex organization in a universe that tends toward entropy. We've also explored the evolutionary perspective that suggests aging may not be a design flaw, but rather an inevitable consequence of how natural selection shaped life. The idea that aging emerges from trade-offs between early reproduction and late survival, between individual cellular immortality and multicellular cooperation. The recognition that different species have found different solutions to the challenge of aging, from the potentially immortal jellyfish to the centuries-old Greenland sharks swimming in Arctic waters. This evolutionary understanding doesn't diminish the value of efforts to understand and potentially slow human aging. Rather, it provides a framework for thinking about which aspects of aging might be modifiable and which might represent deeper constraints of multicellular life. It suggests that extending healthy human lifespan may be possible, but will require understanding and working within the evolutionary logic that shaped our biology. The molecular mechanisms of aging that researchers have uncovered, from protein misfolding to cellular senescence to stem cell exhaustion, represent not just problems to be solved, but windows into understanding life itself. Each mechanism tells part of the story of how complex systems maintain themselves over time, how they respond to challenges, and how they eventually change or fail. The interventions we've discussed, from lifestyle modifications to pharmaceutical approaches to more speculative genetic and cellular therapies, represent humanity's growing ability to influence these fundamental biological processes. Exercise activating cellular stress responses, dietary choices affecting insulin signaling and autophagy, sleep supporting brain maintenance and hormone regulation, potential drugs targeting senescent cells or enhancing DNA repair. These discoveries suggest that aging, while natural and universal, is not immutable. The rate and manner of aging can be influenced by our choices and potentially by medical interventions. This knowledge brings both hope and responsibility. Hope that we might extend healthy lifespans and reduce age-related suffering, and responsibility to use this knowledge wisely. The philosophical questions raised by longevity research remind us that extending lifespan is not just a technical challenge, but a deeply human one. What would it mean to live for centuries? How would extended lifespans change relationships, societies, and individual human experience? These questions don't have simple answers, but exploring them helps us think more clearly about what we hope to achieve through aging research. Perhaps the most profound insight from our journey tonight is that aging is not something that happens to us, but something we participate in. Every breath we take, every meal we eat, every choice we make about how to spend our time influences the aging process. We are not passive victims of time's passage, but active participants in shaping how time affects us. This participation happens at multiple levels. At the cellular level, our lifestyle choices influence gene expression, protein function, and cellular repair mechanisms. At the psychological level, our attitudes toward aging can affect our health and longevity. At the social level, how we structure our communities and support systems influences how well we age. At the species level, our collective research efforts are beginning to unlock aging's mechanisms and develop interventions. Understanding aging as a process we participate in rather than simply endure can be empowering. It suggests that we have more agency in how we age than we might have thought. While we cannot control all aspects of aging, genetics, environmental factors, and chance all play important roles, we can influence many of the factors that determine whether we age well or poorly. This knowledge also brings perspective on time itself. Our human experience of time is shaped by the biological processes of aging. The quickening pace of years as we get older, the way memories fade and shift, the gradual changes in how our bodies and minds function. All of these reflect the underlying biology of aging. Understanding this biology helps us understand our own relationship with time. As you settle into sleep tonight, consider the remarkable fact that your body is actively working to maintain and repair itself. Millions of cellular processes are quietly humming along, fixing damage from the day, clearing away waste products, preparing your body and mind for tomorrow. Your immune system is patrolling for threats. Your brain is consolidating memories and clearing metabolic debris. Your muscles are recovering from the day's activities. This maintenance work continues throughout your life, a testament to the extraordinary sophistication of the biological systems that keep you alive and healthy. That these systems eventually show signs of aging doesn't diminish their remarkable capabilities, but rather reminds us of both the achievements and limitations of evolution's solutions to life's challenges. Sleep itself represents one of evolution's most elegant solutions to the maintenance requirements of complex life. During sleep, many repair processes become more active. Growth hormone is released. The brain's cleaning system activates. Cellular stress responses are fine-tuned. In a sense, sleep is time we spend actively participating in the maintenance processes that slow aging and promote health. So as you drift off to sleep, you can rest in the knowledge that you are part of an ancient and ongoing conversation between life and time. Your body carries within it the wisdom of billions of years of evolution, sophisticated mechanisms for dealing with the challenges that time presents. You are both the inheritor of this biological heritage and an active participant in how it unfolds. The future of aging research holds promise for extending healthy human lifespans and reducing age-related suffering. But even as we work toward that future, we can appreciate the extraordinary biology that already exists within us, the cellular machinery that maintains life in the face of time's constant pressure. Tonight, as molecular processes work quietly within you, as your body engages in the ancient rituals of rest and repair, you are participating in one of life's most fundamental activities, the ongoing negotiation with time that has characterized living things since life began. It's a conversation that connects you to every organism that has ever lived, a shared experience that transcends species and spans the history of life on Earth. Rest well, knowing that you are part of something vast and beautiful, a process that began billions of years ago and continues through you, in you, and beyond you. The science of aging reveals not just how we grow old, but how miraculous it is that complex life persists at all in a universe where most things tend toward simplicity and decay. In the quiet darkness, as your breathing slows and your mind begins to drift, you carry within you the story of life itself, its struggles with time, its solutions to the challenges of maintaining order and complexity, its ongoing dance between preservation and change. You are both a temporary arrangement of atoms and molecules, and a continuation of an ancient process that connects all living things. Before we part ways tonight, let us linger a little longer in the quiet contemplation of time's passage through our bodies, exploring one more dimension of aging that connects us intimately to the cosmos itself. The biological rhythms that orchestrate life at every scale, from the beating of our hearts to the cycling of seasons. Within each of us, a master clock ticks away in a small cluster of neurons called the suprachiasmatic nucleus, nestled deep in the hypothalamus. This biological timekeeper, no larger than a grain of rice, conducts a symphony of rhythms that influence virtually every aspect of our physiology. It responds to the ancient signal of light and darkness, synchronizing our internal processes to the rotation of the Earth. But this master clock doesn't work alone. Throughout our bodies, in nearly every cell and tissue, smaller clocks tick in harmony with the central timekeeper. These peripheral clocks govern the timing of gene expression, protein production, and cellular metabolism, creating waves of activity that rise and fall with predictable regularity. It's as if our bodies contain a vast orchestra, with the suprachiasmatic nucleus serving as conductor, while countless cellular musicians play their parts in perfect temporal coordination. This circadian architecture, our body's way of keeping time with the planet, becomes increasingly important as we consider aging. The precision of these biological clocks tends to deteriorate as we get older, much like an antique timepiece gradually losing its accuracy. The master clock becomes less responsive to light cues. The peripheral clocks begin to drift out of synchrony. The temporal coordination that characterizes youth slowly gives way to a more chaotic pattern of biological timing. This circadian dysfunction may contribute to many aspects of aging. Sleep becomes more fragmented partly because the clock that regulates sleep-wake cycles loses some of its precision. Metabolism becomes less efficient partly because the clocks that coordinate digestive processes and energy utilization fall out of alignment. Even our immune systems, with their own circadian rhythms, may become less effective as these timing mechanisms deteriorate. The discovery of circadian clocks throughout the body has revealed aging as not just a process of wear and tear, but also as a gradual loss of temporal organization. Young organisms are characterized by exquisite timing biological processes occurring in precise sequences, with different systems coordinating their activities like members of a well-rehearsed ensemble. Aging represents a slow dissolution of this temporal precision, a gradual drift toward biological chaos. Yet, understanding circadian rhythms also offers hope for healthier aging. Light therapy, carefully timed to strengthen circadian signals, may help maintain the precision of our biological clocks as we age. Consistent sleep schedules that reinforce natural rhythms may help preserve temporal coordination. Even the timing of meals may influence peripheral clocks throughout the body, potentially supporting healthy aging through better circadian alignment. Consider how the simple act of watching a sunrise or sunset connects you to these ancient rhythms. The light entering your eyes travels to the master clock in your brain, helping to synchronize your internal timing systems with the rotation of the Earth. This daily reset, repeated throughout your lifetime, represents one of the most fundamental ways your body maintains its connection to the cosmic cycles that have shaped life for billions of years. As you age, maintaining this connection becomes increasingly important. The gradual weakening of circadian rhythms doesn't have to be accepted as inevitable. Regular exposure to bright light during the day, darkness at night, consistent meal times, and predictable sleep schedules can all help maintain the temporal organization that characterizes healthy aging. Our exploration of aging would be incomplete without acknowledging the remarkable diversity of aging patterns across different populations and cultures around the world. In certain regions, places researchers have dubbed blue zones, people routinely live to 100 and beyond while maintaining remarkable health and vitality. These pockets of exceptional longevity offer natural experiments in human aging, providing insights into the environmental, social, and lifestyle factors that promote healthy aging. In the mountainous regions of Sardinia, Italy, shepherds in their 90s still tend their flocks, walking miles each day across rugged terrain. The island of Okinawa, Japan, is home to women who regularly live past 100, maintaining independence and mental sharpness well into their final decades. The Seventh-day Adventists of Loma Linda, California, enjoy lifespans significantly longer than the American average, despite living in a modern urban environment. What unites these diverse populations is not genetics alone, though certain longevity genes do appear more frequently in some of these groups. Rather, it's a constellation of lifestyle factors that seem to work together to slow aging and promote health. Regular physical activity, often integrated into daily life rather than pursued as formal exercise. Diets rich in plants, with moderate or minimal consumption of processed foods. Strong social connections and sense of community purpose. Stress management practices often rooted in spiritual or philosophical traditions. The Okinawan concept of ikigai, a sense of life purpose that gets you up in the morning, appears to have measurable effects on longevity. Studies suggest that people with a strong sense of purpose live longer and experience less age-related disease. Similarly, the emphasis on family and community connections in blue zone populations may contribute to longevity through both psychological and physiological mechanisms. Social isolation, increasingly common in modern societies, appears to accelerate aging through multiple pathways. Lonely individuals show increased inflammation, elevated stress hormones, and weakened immune function. Their cardiovascular systems show signs of premature aging. Even their cellular aging markers suggest that loneliness may literally speed up the biological clock. Conversely, strong social connections seem to buffer against many aspects of aging. Married individuals typically live longer than their single counterparts. People with robust social networks show better cognitive function in old age. Grandparents who care for grandchildren often exhibit better physical and mental health than their non-caregiving peers, suggesting that purposeful relationships may have protective effects against aging. These observations highlight an important truth about aging. It's not just a biological process occurring within isolated individuals, but a phenomenon deeply influenced by social, cultural, and environmental contexts. The way we age is shaped not only by our genes and cellular machinery, but also by our relationships, our communities, and our sense of meaning and purpose. The traditional respect for elders found in many cultures may itself contribute to healthier aging. When older adults are valued for their wisdom and experience, when they maintain meaningful roles in their communities, they may age more successfully than in cultures that marginalize or dismiss older individuals. The psychological experience of aging, whether it's viewed as decline or as the accumulation of wisdom and experience, may influence the biological reality of how we age. This cultural dimension of aging becomes particularly relevant as we consider interventions to promote longevity. Technical solutions, drugs, genetic therapies, cellular reprogramming are only part of the picture. Creating environments that support healthy aging may be equally important. This means designing communities that encourage physical activity and social interaction, It means maintaining meaningful roles for older adults. It means addressing the social isolation and ageism that can accelerate biological aging. The study of blue zones also reveals that exceptional longevity doesn't require perfect adherence to ideal lifestyle practices. The centenarians in these populations aren't necessarily paragons of health behavior. Many have smoked, drunk alcohol, or eaten foods that health experts might discourage. What seems to matter more is the overall pattern of their lives: moderate physical activity, strong social bonds, sense of purpose, and generally healthy eating habits sustained over many decades. This observation offers both hope and practical guidance for aging well. It suggests that healthy aging doesn't require perfection, but rather consistency in a few key areas. It's not about optimizing every aspect of lifestyle, but about creating sustainable patterns that support the fundamental biological and social needs that promote longevity. As our understanding of aging has deepened, one of the most surprising discoveries has been the crucial role played by the trillions of microorganisms that live within us. The human microbiome, the collective genome of all the bacteria, viruses, fungi, and other microbes that call our bodies home, changes dramatically as we age. And these changes may both contribute to and result from the aging process. In youth, our microbial communities are typically diverse and stable, with different species of bacteria occupying specialized niches throughout our bodies. The gut microbiome, by far the largest and most studied of these microbial communities, contains hundreds of different bacterial species living in complex ecological relationships. Some produce vitamins that our bodies need. Others break down dietary fiber into beneficial compounds. Many help train and regulate our immune systems. But as we age, this microbial diversity tends to decline. Certain beneficial bacteria become less abundant, while others that may promote inflammation become more prominent. The overall stability of the microbial community decreases, making it more vulnerable to disruption by antibiotics, dietary changes, or illness. This age-related shift in the microbiome appears to contribute to several hallmarks of aging. Changes in gut bacteria may increase intestinal permeability, the so-called leaky gut phenomenon, allowing bacterial products to enter the bloodstream and trigger chronic inflammation. The decline of certain beneficial bacteria may reduce the production of short-chain fatty acids, compounds that help maintain intestinal health and may have anti-aging effects throughout the body. The microbiome also influences brain aging through what researchers call the gut-brain axis. Gut bacteria produce neurotransmitters and other compounds that can affect mood, cognition, and behavior. They communicate with the brain through the vagus nerve and through molecules that enter the bloodstream and cross the blood-brain barrier. Age-related changes in the microbiome may contribute to cognitive decline and increased risk of neurodegenerative diseases. Perhaps most remarkably, research suggests that the microbiome may influence lifespan itself. Studies in laboratory animals show that transferring gut bacteria from young mice to old mice can improve various markers of health and aging. Conversely, transferring bacteria from old mice to young mice can accelerate some aspects of aging. While such experiments cannot be performed in humans, they suggest that the microbiome may be both a driver and a potential target for anti-aging interventions. Maintaining a healthy microbiome throughout life may represent one of the most accessible approaches to promoting healthy aging. Dietary choices have profound effects on microbial communities. Diets rich in diverse plant fibers tend to promote microbial diversity and the growth of beneficial bacteria. Fermented foods like yogurt, kefir, kimchi, and sauerkraut can introduce beneficial microbes and support their growth. Conversely, diets high in processed foods, sugar, and unhealthy fats may promote the growth of harmful bacteria and reduce microbial diversity. Excessive use of antibiotics, while sometimes necessary for treating infections, can disrupt microbial communities in ways that may have lasting effects on health and aging. The connection between the microbiome and aging adds another layer to our understanding of why lifestyle factors like diet and exercise influence longevity. These interventions don't just affect our own cells and organs. They also shape the microbial communities that live within us, which in turn influence our health and aging trajectory. This microbial perspective on aging also highlights the interconnectedness of all life. We are not just individual organisms aging in isolation, but complex ecosystems where human cells and microbial partners age together. The bacteria that live within us have their own evolutionary histories, their own strategies for survival and reproduction, their own responses to environmental changes. Our aging represents the aging of this entire ecosystem, not just the human components. Understanding this microbial dimension of aging opens new possibilities for intervention. Probiotics, beneficial bacteria that can be consumed as supplements or foods, may help maintain healthy microbial communities as we age. Prebiotics, compounds that feed beneficial bacteria, may support the growth of longevity-promoting microbes. More sophisticated interventions, like fecal microbiota transplantation from young to old individuals, are being studied as potential anti-aging therapies. As we near the end of our journey through the science of aging, it's worth reflecting on how modern research has validated many ancient practices and traditional approaches to healthy aging. Across cultures and throughout history, humans have developed strategies for aging well, often without understanding the biological mechanisms involved. Today's research increasingly reveals the scientific basis for these time-tested approaches. The practice of meditation, developed thousands of years ago in various spiritual traditions, has been shown to have measurable effects on biological aging. Regular meditation practice can reduce inflammation, lower stress hormones, and even affect telomere length, potentially slowing cellular aging. The stress reduction that comes with meditative practices may help maintain the precise coordination of biological systems that characterizes healthy aging. Traditional dietary practices from around the world often align remarkably well with what modern science suggests for healthy aging. The Mediterranean diet, the traditional Okinawan diet, the eating patterns of other long-lived populations, all emphasize whole foods, plant diversity, moderate portions, and social aspects of eating. These dietary traditions evolved over centuries of cultural experimentation, preserving approaches that promoted health and longevity while discarding those that didn't. The importance of social connection and community support, emphasized in traditional cultures worldwide, finds validation in modern research on social isolation and aging. The practice of honoring elders, maintaining multi-generational households, and creating meaningful roles for older adults may represent sophisticated social technologies for promoting healthy aging. Even the timing of daily activities in traditional societies often aligns with circadian biology. Rising with the sun, eating the main meal during daylight hours, winding down as darkness falls. These patterns, common in pre-industrial societies, support the biological rhythms that modern research shows are crucial for healthy aging. The integration of physical activity into daily life, rather than separating exercise into discrete gym sessions, reflects a natural understanding of how the human body is designed to move. The walking, lifting, carrying, and varied physical tasks of traditional lifestyles provide the kind of regular, moderate activity that research shows is optimal for longevity. This convergence of ancient wisdom and modern science suggests that our ancestors, through generations of observation and cultural evolution, developed remarkably sophisticated approaches to aging well. While we shouldn't romanticize traditional lifestyles, they often involved hardships and shorter lifespans due to infectious diseases and other factors. We can learn from the aspects of traditional living that promoted healthy aging. The challenge for modern societies is to integrate these time-tested principles with contemporary life. This might mean creating communities designed to encourage walking and social interaction. It might mean developing work patterns that allow for regular physical activity and stress recovery. It might mean rediscovering the social and spiritual practices that give life meaning and purpose as we age. As our exploration draws to close, it's important to acknowledge that despite all the remarkable advances in aging research, fundamental mysteries remain. We still don't fully understand why some individuals age much more slowly than others, even when they share similar genetics and lifestyles. We don't know the upper limits of human lifespan, or whether such limits even exist. We're only beginning to comprehend the complex interactions between genes, environment, and chance that determine how each individual ages. This uncertainty isn't a failure of science, but rather a recognition of the extraordinary complexity of aging as a biological phenomenon. Every advance in our understanding reveals new layers of complexity, new questions to explore, new mysteries to unravel. The more we learn about aging, the more we appreciate how much we still don't know. There's something both humbling and wonderful about this ongoing mystery. It reminds us that aging, despite being a universal human experience, remains unique for each individual. Your particular constellation of genes, experiences, choices, and circumstances creates an aging trajectory that has never existed before and will never be repeated. You are conducting your own experiment in how to age, guided by scientific understanding, but ultimately writing your own unique story of time's passage. This individuality of aging means that while general principles can guide us, exercise regularly, eat well, maintain social connections, find purpose, manage stress, each person must discover their own path to aging well. What works for one individual may not work for another. The interventions that slow aging in laboratory mice may not translate directly to humans. The lifestyle patterns that promote longevity in one culture may need adaptation for others. Perhaps this is as it should be. Aging, after all, is deeply personal. It's about how we navigate the changes that time brings to our bodies, minds, and relationships. It's about finding meaning and purpose across different life stages. It's about accepting the limitations that come with aging while continuing to grow and contribute in whatever ways remain possible. The mystery of aging also connects us to larger questions about life itself. Why does life exist at all in a universe that seems to favor simplicity and entropy? How does the extraordinary complexity of living systems persist and evolve over time? What is consciousness, and how does it change as our brains age? These are questions that extend far beyond biology into philosophy, physics, and the deepest mysteries of existence. In contemplating aging, we're contemplating our place in the cosmos, our connection to the vast history of life on Earth, our responsibility to future generations, who will continue the ongoing experiment of conscious beings trying to understand their own nature. We are both subjects and objects of study, observers and participants in the great mystery of aging. In the quiet darkness, as your breathing slows and your mind begins to drift, carry with you the wonder of what you are, a temporary arrangement of atoms that has somehow learned to contemplate its own existence, to question its own nature, to dream of possibilities beyond its current limitations. You are both a product of billions of years of evolution and a participant in shaping what comes next. The science of aging reveals not just how we grow old, but how miraculous it is that we exist at all. In a universe trending toward maximum entropy, you represent a beautiful, temporary victory of organization over chaos, of meaning over randomness, of consciousness over the void. Your aging is not just personal decline, but cosmic significance, the universe's way of experiencing itself through your particular perspective, your unique combination of thoughts and feelings and experiences. Sleep well, knowing that you are both utterly ordinary, one of billions of humans aging, according to universal biological principles, and absolutely extraordinary, a conscious being contemplating the mystery of its own existence in a cosmos vast beyond imagination. Your dreams tonight will be neurons firing in patterns shaped by millions of years of evolution, creating experiences that exist nowhere else in the universe, except in your sleeping mind. Tomorrow will bring one more day in your ongoing experiment with time, one more opportunity to participate consciously in the ancient process of aging, one more chance to contribute your unique verse to the great song of life. Rest now in the peaceful certainty that you are exactly where and when you belong in the vast story of the universe, aging gracefully through time as part of something infinitely larger and more beautiful than any of us can fully comprehend. Sweet dreams, fellow traveler in time. May your sleep be peaceful, your repair processes efficient, and your tomorrow bright with the continued unfolding of your unique journey through the mysterious and beautiful landscape of life. Rest in the knowledge that you are part of something vast and ancient and ongoing, the endless dance of life with time, playing out in countless variations across the universe, but never quite the same way twice.